is what that is whatever we are supposed to get on that part. Within the Punnett square, I've already explained whatever should be shown there. So I want to invite anyone who has got a question regarding to the first part, which is on the genetics, to ask before we move to the next area. So anyone with a question on the first part, that is on the genetics that we have already covered. Yeyote mwenye ako na swali on that first part? Any clarification on that first part? Any clarification? As I just, I can see within the charts there is a question. Or some of us have not covered genetics. Oh, sorry for that, but I said that I was guiding you. By the time that you will be discussing the details of that, you will be knowing the components of a genetic cross. So all the same, you have learned on those components and the rules that we are supposed to follow when we are drawing those genetic crosses. So for those ones who have covered, within the questions that had given or within the paper that we were doing, so I wait for any other question regarding on that part. You are supposed to explain the actual phenotype ratio obtained from this cross could differ from the expected. Whenever we are making the genetic crosses and we obtain the offsprings, sometimes the chances or those phenotypes of the offsprings that we get may be different from those that are expected because of some of the processes that will undergo within the plant, like now the process of mutation and also the process of crossing over. That is during the process of cell division. We should understand this, that at process 1, there is a transfer of genetic material from one homologous chromosome to another. So that makes it to be slightly different. So the ones obtained during the real or the process of breeding will be different, slightly different from what we normally get when we are doing the cross. Then the disorders due to the non-disjunction, for those ones who have covered that, I only want to guide you that whenever we are writing these defects that occur as a result of the non-disjunction, like now the Down syndrome, the letter D should be capital, Klinefelter syndrome, capital K, Turner syndrome, capital T. So whenever we are writing those disorders, we are supposed to start with a capital letter because they represent the names of the people who first discovered those disorders. And that apostrophe should be there. Just the same way we normally indicate the word Benedicts. We know that Benedict's B should be capital and also the apostrophe before S should be indicated there. So that is all that I can say about this part of the genetics. I want us to move to the next area. Mr. Paul, you can take us to the next part within this document. Yeah, that is section number 2. Yeah, that way it's okay, thank you. So within this part, it is questions on the experimental setup. We do not miss questions on the experimental setup within the exams. They are always there within our exams. And whenever we are dealing with these questions on the experimental setups, one thing that we are supposed to get is this, that you should understand the factor that is under investigation. I've listed here some of the common areas that are normally tested on the experimental setups. Like now, the photosynthesis. Photosynthesis, here, you normally get many questions on the photosynthesis that are those experimental questions normally cover on the factors or the conditions that are necessary for the process of photosynthesis. Like now, is carbon 4 oxide necessary for the process of photosynthesis? You normally get that experiment that has got a polythene paper that has been enclosed with sodium hydroxide pellets. We know this, that the sodium hydroxide pellets are responsible for absorbing the carbon 4 oxide. So they will be able to take a plant that has been kept in darkness for 48 hours, then place it within the sunlight. One of the leaves covered with a polythene paper that is transparent but containing within it sodium hydroxide. Left for some time within the sunlight, then the iodine, meaning the iodine tests, all tests for starts will be carried out within the leaves. That one which was exposed to the sunlight and another one that was enclosed within a paper containing the sodium hydroxide. That is one example that we have there. Another one that is on the respiration. Here on the respiration, there are many questions that we normally get. Like now, the glucose solution that has been immersed within a water bath, maintained at temperatures of 37 degrees Celsius, then connected to a test tube containing lime water. That is an example. You can also get another one containing glucose solution that has been boiled, cooled, then yeasts added to it, then oil, then a delivery tube has been connected between that setup to lime water again. You can also get another one containing germinating seeds that have been placed within a thermos flask. Then a thermometer has been inserted within that thermos flask. Those are some of the experimental questions that we normally get. Growth and development, you can get one that is having germinating seeds again, then which will be investigating the different conditions that are necessary for the process of germination. Like now, water is responsible or it's required. We have got the optimum temperatures. Whenever we are indicating the temperatures, we should indicate optimum temperatures, but not temperatures alone. Then we also know that enzymes are also required for this process to take place. Oxygen is required because it's the one that will be used for the respiration to oxidize the food stored within the seeds to provide energy for the process of germination. In other setups, you will get that there is a removal of or there is a presence of the pyrogaric acid. The pyrogaric acid is normally used to absorb the oxygen, and we know that the oxygen is required for the process of respiration to give energy for the germination process. Gaseous exchange is also another part that is normally tested in form of experimental setups. An example of this process, you may get like now a plant that has been placed within a certain point, and the gas that is being produced is collected. A gas that is being produced is collected and tested. Like now, you can place a plant within the light. We know that if you enclose it within a transparent polythene paper, the process of photosynthesis will continue to take place. Then after that, you can be able to take that gas, investigate that gas. Or as well, you can be able to place the same plant within a dark a dark environment, collect the gas that it will produce, and investigate on that. That is, or those are some of the examples of the experimental questions that we normally get within the exams. Whenever we get the questions on the experimental setup, the main thing that we are supposed to get here is that you should get the aim of the experiment. We pick the aim of the experiment from the factor under investigation within an experiment. Like now, if you see a plant that has been covered, a leaf of a plant that has been covered by the aluminum foil, you will understand that it is light that is being investigated. But we do not indicate light just that way because we have got different aspects of light. In case you cover the polythene or they live with an aluminum foil, in this case we say to investigate the effects, to investigate the effect of light intensity on the rate of photosynthesis. So that aim should start with the word to investigate, to find out, to check, to test that way. But the factor that is under investigation should be listed within that question that you are having, or within the aim of that, experiment. Apart from the aim, the other thing that we are supposed to get whenever we are dealing with the experimental questions is the observations. Which observations do you make whenever you have carried out that experiment? I use the one for the photosynthesis. For the experiments involving the photosynthesis, we normally carry out the starch tests. The main reagent that we use for the starch test is iodine. And the color of iodine is normally brown. And in presence of starch, this iodine changes to blue-black. Normally, we should say iodine solution. Be specific. The color of the iodine solution changes from brown to blue-black. That is in presence of starch. So whenever we are responding to those questions, whenever we are giving the observations, we should understand the correct way that we give our observation by indicating, like now, for these experiments involving photosynthesis, whether the iodine solution is changing from brown to blue-black, or the color of iodine solution, the brown color of iodine solution, does not change. For those ones that are involving respiration, whenever we are carrying out the process of respiration, like now, if it is the seeds that are germinating, they will generate energy in form of heat. That is why we normally place the thermometer there. So you are supposed to say the temperature increases. The temperature increases. And how will you be able to know that the temperature has increased? Is taking on the reading on the thermometer. If it increases, it shows that the temperature has increased. That is an observation. The gases that are being produced, like now, the carbon 4 oxide, there is a delivery tube connecting between the substances where all the solutions where this respiration will be taking place to the lime water. So we expect the color of the lime water or the lime water to form a white precipitate. So a white precipitate is formed. That is the correct way that we are supposed to indicate whenever we are using the lime water. Growth and development, also for these ones, we are supposed to indicate whether the germination process occurs or does not occur, because we can be able to note that the germination has occurred if there is sprouting or the formation of the plumule and the radicle, or the formation of the shoots and the roots, based on the conditions that have been given there. So observations should be clearly indicated. We normally obtain the explanations of the processes that are occurring within the experimental setups from the observations that we have made there. So that is where we normally get those explanations. So I will not be specific on the explanations that we can be able to give, but for those ones who have already done question number 2, in the question paper that I had given, Mr. Paul, you can take us to the sample paper, kindly. Sample paper that I had sent, question number 2. So as Mr. Paul is displaying that, I can say this, that in almost every year, there is a question on the experimental setup. Like now, in the year 2024, last year, in the paper 2, we had a question on the photosynthesis, experiment on photosynthesis. In the year 2023, we had a question on photosynthesis. There is an experimental question on photosynthesis and gaseous exchange. In the year 2022, kulikuwa na ya gaseous exchange. In the year 2021, there was an experimental question on gaseous exchange. In the year 2020, kulikuwa na question on germination, growth and development. That is from that topic of growth and development. So you can see, for the last 5 years, the questions on the experiments do not miss. So we are supposed to ensure that we follow those guidelines that I've given. Those aims, observations, the explanations, and the applications of those processes that normally occur within those experiments are normally given within our books. Whenever we are doing revisions, there are those questions on the practical activities. If you go to your KLB Form 1, like now on this photosynthesis, kunakuwa na practical activity. If you relate those practical activities that have been given there, they are the same ones that are normally tested or that are normally given within these questions that are normally tested within the KCSE. So get this, that you are supposed to go through those practical activity, understand what is being investigated and the questions that are involved within that. Mr. Paul, within that part, you kindly display for us, there is a question too within the sample paper to that I'd sent. Meanwhile, I can be explaining what that question was involving. So the question was about a solution that was connected to an organism that was in a flask. It's a mouse that is in a flask that is airtight. Then it's connected to another solution that is connected finally to the filter pump. So if you look at that document, you will get that the process that is happening within this mouse, which is in a conical flask, that leads to production of a gas that changes the color of that solution D that we are having there. So we know that this rat, there is the process of respiration that will be taking place within it. And when the respiration process occurs, there is production of a gas, that is carbon 4 oxide. The carbon 4 oxide will be the one that will be responsible for the changing of the color of this lime water, that is solution D, which forms a white precipitate. So the white precipitate that is formed is an indicator that the carbon 4 oxide has been produced there. Then you can get this that what they normally ask is the objective. Like now in this one, mulikuwa umeulizwa you identify the process being investigated. Of course, the process is respiration. Some students can argue that the process occurring here is a gaseous exchange, but we should see if we are mentioning about gaseous exchange, the direction in which these gases will be moving from where to where. But because it's formation of a specific gas, so we are supposed to a specific gas that is changing the lime water. So that is why we prefer or we give the correct answer for this question as respiration. Yeah, it's that paper, Mr. Hall. Question number 2 can take us to question number 2. Yeah, question number 2, it's there. Yeah, this one. Already I've explained to the students here that these two solutions, one is containing sodium hydroxide to indicate sodium. It should be sodium hydroxide. The reason why we are having sodium hydroxide within this first part is to ensure that there is no carbon 4 oxide that will be passing to this mouse within the conical flask. So inakuwa eliminated yote from this first flask. So kwa hii mouse, a process is occurring within it that is responsible for the change of color of this solution D. Hata naye hatukuwa tumeindicate vizuri, inastahili kukuwa solution D. So the process that is being investigated here, I'm saying it is the respiration. So let's move to the questions, Mr. Paul. To the question so that I can be able to take them through the first question. Yeah. So you identify the process being investigated. So I've said the response that we're supposed to give under that is respiration. Why? The carbon dioxide that is being produced that will be responsible for the change of color of solution C is the one that the respiration process is the one that will be responsible for the production of that gas. So it's the process occurring within the rat, not all the mouse that we'll be looking at. Name solution C, of course, because we are investigating for the gas produced, that should be lime water. The chemical name for lime water is the calcium hydroxide solution. So if someone indicates that way, it will be correct also. But for biology, you just mentioned lime water. We are not very keen on the chemical name. For part C, state the observation expected at the end of the experiment in tube A. Of course, from whatever we had within that part, the first tube is the one that we refer to as the tube A. And we do not expect the color to change. Within the atmospheric air, very little, because above that solution, kuna sodium hydroxide that normally take all the carbon 4 oxide. So hatustahili kukuwa na any change in color of this substance. No change in color. Whenever we are writing the observations, we are not supposed to indicate no observable change. You are supposed to say either the color of the solution is retained or the solution remains colorless. Because we know that this calcium hydroxide or lime water is normally a colorless solution. So it remains colorless, or as well you can say no change in color. But we are not supposed to say that it it's not no observable change. It's not correct for us to say no observable change. Then the next part, take us to the next part, Mr. Paul. Yeah, tube B, although there was a confusion with the letters, tube B is the last tube after the respiration has occurred. Then we know that the process of respiration gives carbon 4 oxide that leads to the formation of the white precipitate. So we indicate two white precipitate. Accounting for the observation made in A, no carbon 4 oxide gets into the solution because the carbon 4 oxide was absorbed by the sodium hydroxide. So before it gets to the calcium hydroxide, the sodium hydroxide penetrates within that entry, takes all that carbon 4 oxide. To be why there is formation of a white precipitate, it's because the organism respires. And when it undergoes the process of respiration, it produces carbon 4 oxide responsible for the color change. I would also like to get any question on these experimental questions before we move to the next area. So any question on the experimental parts? Any question on the experimental parts? Silence means that we are okay. So if there is any question, just indicate it within the chat. I will be able to respond to that question as we continue. So you can just type that question within the chat. I will be able to go through it as we continue with the next part. Back to the initial document, Mr. Paul. The next part, as we get there, points.
Just before you get there, Mr. Makori.
Yes.
I would wish to know whether Noela is in this class, and I will also wish to know whether Sanayan is getting it. I need to hear from them. They are our new students. I need to know whether they are doing well. Noela, are you here? Maybe you may have entered with another name. I'd also wish to know whether Sanaya from Kipsigis Girls. Sanaya,
I'm good.
if you are still okay,
I'm
that
good.
is okay. Thank you. Noela, are you still here? AC, how are you? AC, how are you? I think it's okay, Mr. Makori. Let us just proceed. I will look for the two.
Yes, thank you. So I can see there from TK Onjonga, is there any control experiment, and if so, what can it be? So there is a control experiment for this. The organism that is placed there is alive and is carrying out the process of respiration to give the carbon 4 oxide. So the control experiment is either we have a dead animal, a dead mouse within the conical flask, or as well we can miss to have any animal within this part. So we make the setup the way it is without the mouse within the conical flask, or we use a dead mouse within that animal. The reason why we do not prefer a dead mouse, because it can be decomposing. The decomposers can also lead to the production of the carbon 4 oxide. So the best ni kukua na conical flask, set up vile with a conical flask that has no organism for that. Because we know that the control experiment is for comparison. So we will know that the carbon dioxide was produced by the orgasm when the respiration was taking place. But no carbon dioxide is going to be produced because no orgasm is there and no respiration will be taking place within this part. Thank you. So we move to the next part. Oh, there is another question. I can be able to go through it. Yes. AC is asking, why is it all not understood why it's respiration? I can explain it once again. Within the setup, tuko na sodium hydroxide, we have an animal, we have another boiling tube with the lime water or the calcium hydroxide. Of course, whenever we are going through with the students on these questions, they normally, for them, they feel like we should say it's gaseous extents. But why we do not accept the gaseous extents, it's because we are supposed to show the direction in which these gases are moving from and to which place. So we do not accept gaseous extents. It is the respiration because what is producing this carbon dioxide within this organism? It is the respiration process. It is not the gaseous exchange process. It is the respiration. The respiration produces the carbon dioxide that is going to be transferred. Yeah. So unaona hapa hii organism yenye tuko nayo within the screen, that mouse, the respiration occurring within the mouse is the one that produces the carbon dioxide. So that is whatever we are getting within that part. Lucy is asking, she's not getting the, I can go through that. I hope, Lucy, you are getting it now. Oh, we alikuwa ni AC. So the process that is occurring within the organs that we look at. Then I respond to, oh, Lucy anauliza, the observation in test tube A or within the first test tube. Look at that document. Look at this document here. Uko juu, yeah, hiyo A, yeah. Uko juu kuna sodium. I've said it should be sodium hydroxide pellets. Sasa kama ziko at this point, sodium hydroxide, kama hii ni sodium hydroxide within this point, inamaanisha the air that is getting into this test tube A does not contain carbon dioxide because that carbon dioxide has been taken by this sodium hydroxide within this part. So that is why haiko within, or the color of this solution A does not change or remains colorless. Yeah, is it correct to use a negative observation? Like now, is it correct for one to say there is no formation of a white precipitate? That is, I think that is what it means by a negative statement. That is the expected color. So if you indicate that it will not be formed, it will be okay. Kuna mwingine mwenye ameuliza let me go in sequence. Please account for test tube A again. Test tube A, I've said already that Sylvia, that test tube A, the accounting for that is that the carbon dioxide from the air is absorbed by the sodium hydroxide at the entry point, huko juu, at the entry point. So no change in color of the calcium hydroxide solution. Hiyo yenye iko solution yenye iko huko chini ni calcium hydroxide. Above that ni sodium hydroxide. Sodium hydroxide inabsorb carbon dioxide. Calcium hydroxide in attest for the presence of carbon dioxide. So ina confirm that there is no carbon dioxide that moved into the solution. when should the answer be gaseous exchange? That is a good question by Teresa. So the can be gaseous exchange if maybe we were to show within a membrane movement of a gas from one point to another. A gas that was initially within point A is detected in a point B. That is when we can be able to show that that is gaseous exchange. But in this one, there is no way that we can be able to show that the carbon dioxide was initially within this organism and it has moved to this boiling tube that we are having there. But we can conclude that the respirations that occurred within the mouse led to the production of the carbon dioxide that we are observing there. So kama tungekuwa na gas at that mouse and we detect its movement from that point to another point, that is when we can be able to say that it's gaseous exchange that is occurring. Do we use or do we say to act as a control experiment or it's a control experiment? It should be a control experiment. We do not say act. When we are saying act, it means that that experiment is it's being carried out instead of another one. It's replacing another one. So we should not say act, but we say it is a control experiment. So the second part of the statement is the correct way that we are supposed to give it. So that is what we are supposed to get on those parts that we have already asked. If there is more or another question, I will be able to respond to that. So you can be able to send your question within the chat section. I will be able to go through it. Back to the original document, Mr. Paul. We move to the part of the cell physiology, and I will summarize the cell physiology within the 15 minutes. The cell physiology. Questions on the cell physiology rarely means within the exams. In the year 2024, the question on cell physiology was there. In the year 2023, it was not there. 2022, it was there. 2021, it was not there. 2020, it was not there. So last year, we had a question on the cell physiology, and that question, most of the students didn't score the correct marks or good marks on that part. So on this cell physiology, the first thing that we are supposed to get anytime that we are on this part of the cell physiology is that we are supposed to identify the type of physiological process that is taking place. We later go to the document that we are having, but we know the common types of processes that we normally have. Like now we have got the osmosis, we have got the diffusion, active transports rarely given within the experiments that we normally get, because it's difficult to show how the energy will be provided for that physiological process to occur. So the most common ones that we normally get are the ones involving the osmosis and the diffusion process. Now, on the osmosis, we are mainly supposed to see those experiments that are involving the movement of the water molecules. If an experiment involves the movement of the water molecules, then it means that that is the osmosis process. But if it is another form of particle, then it will be the diffusion. Most cases where you normally see distilled water, either a cell has been immersed within distilled water, or you may get a plant tissue, or a fisking tubing has been immersed within distilled water. You automatically know that whatever is being investigated there is osmosis. Experiments on diffusions. The common one that we normally do is the one that involves the iodine and also the starch. You take the iodine, you place it within a fisking tubing, then you immerse that fisking tubing containing the iodine solution, to be specific, iodine solution. You immerse it in a solution containing starch. Then you observe whatever is going to happen with that mixture. Of course, huwa tunaona the starch mixture changing to blue-black. And we normally say that the iodine molecules are the ones that diffuse from the fisking tubing into the solution that is contained within the beaker. So because it's involving different particles, then we refer to that one as the diffusion. There are other experiments whereby you get that, like now unaweka enzyme. There is one that I saw somewhere whereby unaweka enzyme in a test tube, not a test tube, but a fisking tubing. Then that enzyme is placed within a food substrate that it acts on. Like now, unaweka starts with the diastase enzyme, then you immerse them in a solution that is containing a Benedict solution, or a solution of Benedict solution in a beaker that is immersed in a water bath. In this case, the water bath provides the optimum temperatures for the action of the diastase enzyme. At the same time, it will be able to provide the required temperature for the change of the reducing sugars, or to detect the reducing sugars within that setup. So that process that will involve the movement of these glucose molecules after they have been broken down into, or from starch into reducing sugars, then move across the fisking tubing into the Benedict solution that will change that Benedict solution. It will be the diffusion process that will be taking place there. So I wanted to differentiate those experiments that involve the diffusion process and the osmosis. Ukiona anywhere kuna distilled water, most likely hiyo ni osmosis. If it is involving another particle apart from water, that process that will be taking place there will be diffusion. So take note of that. Whenever we are giving, or whenever we are dealing with questions on the cell physiology, we are supposed to be keen on the observations that we make, especially if we are using the fisking tubing. We should check on the color change, if the substances that are being involved are colored, or the increase in size. There is this question here whereby unapewa glucose solution or a sucrose solution within a fisking tubing immersed in distilled water in a beaker or in a trough. Whenever you are giving the observations, the contents within the fisking tubing are the ones that contains in a amount, but the total amount within the container, let's say within the beaker, they do not change. They do not change. Someone is asking, I mentioned the second function of the water bath. I repeat, nilikuwa nimesema tumeweka starch and diastase enzyme in a fisking tubing, then we immerse them in Benedict solutions in a beaker that has been immersed